Hello. Welcome to another lecture on our ongoing series within neurobiology. We've been covering aspects of the sensory systems, starting with the visual system. So if you've missed those two lectures, I've got one on the eye and then one on the central visual pathways. And today we're going to be actually covering two different sensory systems all in one video. We're covering both taste and smell. So why don't we get to it? Now, the thing is, one thing we haven't discussed quite yet within this series is the facial nerves. There are a series of nerves that come off of the brain that innervate various aspects of the face. We refer to these as the facial nerves, and there are 12 of these, and you can find them in, I believe, really any vertebrate, and they're in essentially the same order across all vertebrate brains. Here we are looking at our trusty dusty human brain. We're looking at the ventral aspect of it. So this is if you would take a brain out of a human brain and then flip it upside down. And this is what we're looking at. And we're pointing out all the different facial nerves. I'm not going to go through them all today, but we are actually going to be talking about nearly half of them. We're going to be talking about 5 of them. I'm going to mention that we have discussed one previously, which is the optic nerve. This is the nerve that carries sensory information from the eyes to the brain. So we're not going to be discussing that nerve other than what I just said. And you can look at the last 2 lectures to see more about the optic nerve. We will be discussing these 3 nerves: the facial nerve, the glossopharyngeal nerve, and the vagus nerves. Portions of these nerves innervate aspects of the tongue. So we'll be discussing those. And then we will finish by talking about the information that comes in through the first facial nerve, which is the olfactory nerve. This carries our sensation of smell, and it is shown here in the human brain. This would be the olfactory bulb. And actually, this would be the olfactory tract, to be honest. The olfactory nerve would be the information coming from the olfactory epithelia to the olfactory bulb. And then the bulb does its own processing, and then this would be the olfactory tract. Still the primary olfactory tract. But we'll get into what I'm getting into parsing some of the details. Don't want to give you too many spoilers up front. So let's talk about taste. We refer to this as gustation. Gustation is the sense of taste. When we put something in our mouth, it's going to have a taste to it typically. And it's going to fall into 5 basic categories within human beings. It is saltiness, sweetness, sourness, bitterness, and umami. So let's go through these, discussing them a little bit. So saltiness is found in many salts. This includes almost entirely sodium and chloride. We're going to need sodium and chloride for various processes within the nervous system, as we've talked about, as well as a lot of other things that's important that for what our bodies need. Electrolytes. It's what bodies crave. And that's where we're going to find, that's why saltiness and why we have a taste for saltiness, right? Table salt. Sweetness. This is going to be chemicals such as fructose, sucrose. Artificial sweeteners do activate the sweet receptors, which is why they can give us the sense of sweetness without giving us the calories. And then there are other certain carbohydrates that can trigger sweetness, but it's typically going to be, like the sugars. We have sourness. Now, there are many different types of sours, types of foods. Some of the classic ones, of course, are lemons, limes, grapefruits. This is going to be detecting acidity. Citric acid is one example. Bitterness is things that, so we have lots. Bitterness is as a complex taste form within humans. There's a lot of different subtleties and varieties to bitterness within humans. It's even more complex than some other animals. But it's going to be caused by certain kinds of ions, such as potassium, magnesium. Other chemicals can affect bitterness. And then we got umami, which is Japanese for delicious. This is a taste that was originally identified in Japan, so they got the right to name the taste receptor, and so we call it umami. And it's defined by binding of the amino acid glutamate. We'll get into it. An example of this is MSG, which some people claim to be allergic to. I have a hard time understanding how they could possibly be allergic to it, since it is just a simple amino acid. I don't know. Maybe. It's monosodium glutamate, which means it's a glutamate with a sodium attached to it. It comes in a crystal. But it provides sort of a meat flavor. And so when we eat meats, it's going to be filled with all kinds of proteins. If we eat other things that are protein rich, such as beans can give sort of a protein type flavor to it. And it's mostly because it is mostly protein, and it's going to be the glutamate that's doing that job. So with this limited number of basic tastes, how do you think we perceive all the countless flavors of food and drink? Well, it's going to be three factors that are going to feature into this. One of which is that foods activate distinct combinations of taste receptors. So it's not as if we put something into our mouth and it's only going to activate sour, or it's only going to activate umami. It's going to be a mix of salt and sweet, umami, sour, bitter. All these things kind of get activated, especially if it's a well-prepared meal. Distinct flavors can also occur due to the combination of not just taste, but also smell, as we get into. Smell offers then a lot more flexibility than just these 5 distinct categories of taste. Then, taste isn't just the chemical interaction that happens in our mouth. There's also texture, there's temperature. They contribute to the sensation of taste, sometimes pain, related to whether it's actually hot, or spicy flavors such as capsaicin. We'll talk a little bit more about capsaicin when we get into temperature sensation. So the organs of taste that we depend upon is primarily done through the tongue. Although there are other regions within our mouth that do have taste receptors. Within the pharynx, the palate, and the epiglottis, they do have sensitivity. So it's not just exclusively the tongue, but most of it is located on the tongue. Taste sensing cells are found on taste buds that are distributed across the surface of the tongue in these other regions. And when we are talking about taste stimuli, we refer to them as tastants. So I'll be using that word throughout this presentation. They are non-volatile. They are hydrophilic molecules that are soluble in saliva. So non-volatile means that they don't typically go up in the air and you can't really smell them. Hydrophilic meaning that they like water, and so therefore they are soluble in saliva. The perceived intensity of taste is usually directly proportional to tastant concentration. So the more of the tastant that you can have in a piece of some kind of food, the more intense that taste is. So if you have like a particularly sour candy that's very high in citric acid, it's going to activate a lot of sour receptors, and something like a lemon head or something. But other candies may have just a little bit of sourness to give it a little bit of a bite, but it's not going to dominate quite as much, because then it's going to be more the sugars that you're going to be tasting, since there's more space within the candy for sugar and less for the sour. And so you activate just less of the sour, and it's all related to concentration. So concentration, it's a graded change that can occur here. Now, there are not distinct areas of sensitivity on the tongue. So one thing that you may have seen is a map like this, with bitter at the back and sour on the sides and salty and sweet in the front, that there are these very specific regions. And there are people who even make labs sort of based around testing these different regions, and some sort of basic sort of psychology or neuroscience type labs. It is true that there is general specificity in that there can be more higher concentrations of receptors in certain regions, but there's enormous overlap. So this is what it actually really looks like. So for instance, sure, it is generally true that we have a higher concentration of bitter taste receptors towards the back of the tongue than we do towards the front. But we do have bitter receptors at the front. You can taste bitter things in the front. You're just going to have greater sensitivity towards the back. Same goes for sour, except that those receptors are a little bit more concentration on the edges of the tongue than in the center and towards the back. Salty is directed a little bit more towards the front, but of course there are some receptors towards the back. The same goes for sweet. Sweet is generally located towards the front of the tongue, and less so in the back. Umami is also spread throughout the tongue as well, with higher concentration towards the front. That's what these little yellow dots are supposed to indicate. The sweet is the red. So yeah, there are regions of sort of sensitivity, but you can find these receptors across the entire tongue. It's not as if we've got like a specific spot where you're only going to find sour receptors or you're only going to find sweet receptors. This is wrong. Forget it. It is not real. This is what you want to know. So let's talk about the organs of taste. So as I've said, we've got these tastants, the chemicals that can be found in foods that we're going to put in our mouths. They are detected in the receptive specializations called papillae, which contain taste buds and taste cells. So here we're looking at this dude sticking out his tongue, sticking out really, really far. And what we can see on the surface of the tongue are all of these sort of bumps. And these are various papillae, okay? So within papillae, if we zoom in a little bit more, what we're going to find is that these are bumps that have folds that go into our tongue like this. So if we're zooming in and we're looking at a cross section, this would be the papilla. So it'll be like this is one papilla. This is going to be the little bump that's going to sort of be standing out. But believe it or not, there's this infold that goes into the tongue. And inside that infold is where we're going to find taste buds. So if we can zoom in here, we look a little closer, this is what that's going to look like. So we're going to look at that a little bit more closer on the next slide. But for now, I want to just say that each papilla has anywhere from 1 to several hundred taste buds that contain the neuroepithelial taste cells. This is an example of one taste. No, I take that back. There are several taste cells here. So we've got 1, 2, 3, 4, 5 that we can see here with the taste pore. And each person has anywhere between 2,000 and 5,000 taste buds. But this is like the kind of the average. So something like, I don't know, probably 80% of everyone has somewhere between 2,000 and 5,000 taste buds. But there are people who have very few taste buds. They can have as few as 500. And then there are super tasters. People that, for whatever reason, have an acute sense of taste. And it is now known that those people, it's not just in their head, that they in fact have many, many taste buds. In fact, they can have as many as 20,000 taste buds, more than 4 times the upper range of what is typically found in an adult human being. So if we zoom in on this perspective a little bit more to get a sense of the different kinds of taste cells that we can see within the where they accumulate inside the papilla. Basically, there's going to be an apical end. That's going to be the end that's facing towards where the saliva is. There's going to be what's called a taste pore. That basically that's the tiny little hole that allows the mucus to interact with the microvilli, which is at the surface of the taste cells. So this would be here. This is the taste cells turned on the side. This is with it facing up. And, so if we're zoomed in here, we're looking at the taste bud right here. And here's the taste pore. And here are individual taste cells. Now, within here, there are other kinds of cells. One very important kind of cell is called a basal cell. These taste cells, they don't last forever. They do sort of get replaced over time. They'll die and then get replaced with the basal cells, which undergo proliferation, and then they differentiate into a specialized taste cell. The receptors are typically found in the microvilli, so they're going to be way up here. And then we're going to talk about the receptors for these cells, how they work, and then the types of neurotransmitters that we can find within the taste cells, the 5 different kinds of taste cells, 2 of which use serotonin as its neurotransmitter, and then the 3 others use ATP as its neurotransmitter. So the basic mechanism of taste transduction is that we have taste stimuli, the tastants, be it salts or acids or other more complex molecules. They will act on a receptor, of course. Some of them will act directly on ion channels. Others act on G protein coupled receptor mechanisms, as we'll get into. And then after that, we're going to have depolarization that occurs within the body of the cell. So this is happening in the microvilli. There could be if it is interacting there is depolarization of the cell membrane. This is true for all the taste cells. This is going to be largely driven by an influx of sodium and calcium. And then we, especially with the G protein coupled mechanism, we've got intracellular release of calcium as well that allows for the release of neurotransmitter. In this example, we're showing serotonin, but ATP is another one. So we're going to get into it. So let's talk about saltiness first. Salt, table salt, is just sodium chloride. Sodium is already the ion that is typically used to allow for depolarization of the membrane. And salt taste cells, since it's using table salt as its tastant, it basically takes advantage of the fact that we're going to have an increase in sodium concentration inside of the saliva, just outside of the cell. So there's going to be an increase in sodium concentration. This creates an even greater electrochemical gradient. So we have an increase in the amount of sodium that flows into the cell. This results in a membrane depolarization. Very simple sort of mechanism. It triggers voltage-gated sodium and calcium channels to open, which then triggers neurotransmitter release. And in this case, serotonin is then released onto the postsynaptic gustatory afferents from those axons. Sourness is similar to salt in some ways. It's not sensitive to sodium ions per se. Instead, what it is sensitive to is hydrogen, except that the hydrogen here acts in two different ways. So hydrogen can, these sour cells have potassium channels that are sensitive to pH. And that if the pH increases in the saliva, it will close those potassium channels. And remember, potassium is high on the inside of the cell. If these channels are closed, then we have less of the leaky potassium that's flowing out. And therefore, that's going to reduce the outward current. And hydrogen ions are positive. There are proton channels in these cells. So if the concentration increases here, we have an influx of hydrogen into the cell. This is positive ions flowing into the cell, lack of positive ions flowing out. It depolarizes the membrane. So instead of it's being held in a hyperpolarized state, it becomes depolarized. It activates voltage-gated sodium and potassium, calcium channels that act on the serotonin receptors, releasing neurotransmitter onto the postsynaptic cell. Now, the next three types don't use ion channels as their major mechanism for detecting the tastants. Instead, they use G protein coupled receptors. And these G protein coupled receptors are dimers for each of them. So it's going to be the combination of the two dimers that allow for the taste to be picked up. Bitterness is the most, you could arguably argue, the most complex form of taste in the human body. This is because we have around 25 different types of bitter receptor proteins. So it allows us to distinguish subtlety of different bitterants. And this is because one of the major functions of detecting bitter is that it's a poison detector. And so, we've evolved the mechanism to have a greater sensitivity to lots of different bitterants because, well, there's a lot of poisonous things out there. So let's walk through how this works. Basically, bitter tasting foods, they bind to some combination of two G protein coupled receptors that are bound to each other. This activates mechanism within the cell. Specifically, it activates the enzyme phospholipase C, or PLC. So this becomes activated, right? G protein coupled mechanism. The bitterant will bind to these receptors, activates the G protein. That leads to the activation of PLC. PLC stimulates the production of IP3. It converts PIP2 into IP3. So intracellular IP3 increases. This is a second messenger system. It acts on the endoplasmic reticulum. It triggers the release of calcium stores. So calcium that is held inside the endoplasmic reticulum is now released. This then also opens up sodium channels. IP3 can do this as well. So we've got an influx of sodium channels. This combination of events, calcium plus the influx of sodium, opens up the pores that allow ATP to flow out of the cell. So you have ATP acting as a neurotransmitter, which acts on specialized receptors on the specialized gustatory axon afferents that are used for bitterness and sensing bitterness. Sweetness operates in a similar way, except that it has only two G protein coupled receptors, T1R2 and T1R3. And so therefore, And it's the combination of these two. They have to be together. And it's just these two. So we don't have 25 to pick from like we do with bitterness. It is just these two because they will pick up, sucrose, glucose, fructose, and then they are also sensitive to artificial sweeteners like aspartame and the other ones, right? So they will bind to this combination of receptor T1R2 and T1R3. So those cells that are expressing those two G protein coupled receptors will allow for us to detect sweetness. And then the pathway is the same intracellular pathway, other than the fact that it's these specialized receptors. It's still the same pathway that's intracellular as we just saw for bitterness. So I'm not going to walk through that again. But if it's the same pathway inside a cell that allows us to taste bitter and sweet tastes, how do we not confuse the bitter tastings from the sweet ones? What do you think? Well, it's because the receptor proteins for each type, bitter versus sweet, are expressed in different taste cells. And then those cells connect to different gustatory axons, right? So some gustatory axons, if they're bound to this combination of receptors, those gustatory axons, if they're receiving ATP, is telling our brains, oh, there's something sweet in my mouth. And then if you have the combination of the bitterant taste receptors, they're going to be bound to bitterant gustatory axons that is going to convey bitter taste. So ATP released onto those gustatory axons says, no, this is a bitter thing that I have in my mouth. Maybe I don't want to eat it or whatever. And then umami. So this is, again, another G protein coupled mechanism, except that it uses the combination of these receptors, T1R1 and T1R3. Okay, so different than sweet. So and in this case, if we have it's used to detect amino acids like glutamate. So monosodium glutamate will bind to these receptors, and then that activates that same mechanism that we just talked about for sweetness and for bitterness, allowing for the release of ATP. And then, once again, how do we not confuse umami and sweet and bitter tastes? Once again, the receptor proteins for each type are expressed in different taste cells. And then those cells connect to different gustatory axons. So cells that express these receptors are going to be connected to gustatory axons that carry the information saying that this is protein tasting, and that when those axons are activated, so all you would need to do is just release ATP onto those specific axons. And if you did, then you would be telling the brain, oh, well, I've got protein in my mouth and this is something that's like savory, right? So let's go over the central taste pathways. So the incoming taste signals, they activate different parts of the tongue. And believe it or not, the tongue is innervated by two different facial nerves. We've got the seventh facial nerve and the ninth facial nerve. So we've got the facial nerve and the glossopharyngeal nerve that innervate the foretongue and then the latter part of the tongue. And then the vagus nerve has taste receptors in here. It's going to be mostly highly concentrated in bitterness, but there are other taste receptors in there. This is behind the tongue. It's largely areas around the epiglottis that gets innervated by the vagus nerve. But these three nerves all convey taste information up into the brain. They synapse onto the nucleus solitary tract of, this is called the gustatory nucleus. It is in the medulla. And then from there, it projects up to the thalamus, specifically the ventral posterior medial thalamus, nucleus of the thalamus. And then that information then gets conveyed to other parts of cortex. So from here, so we got the afferents of the solitary tract, synapsing onto the insular taste cortex. And from there, that information can be conveyed up to the frontal lobe. So that's just the pathway. I don't expect you to have all this memorized, other than the fact that we have three cranial nerves that innervate the parts of the tongue. They convey information up to the medulla and then to the thalamus. So this, I do want you to know that it goes through the thalamus. and then up to the cortex. So again, like the different taste signals that get activated, it's going to be depending upon the part of the mouth that is sensed, and the different cranial nerves. They're going to carry information in very specific ways. And then they synapse up into the thalamus. The solitary tract is also important for this. It's conveying all kinds of information, not just gustatory information, but it's involved in swallowing and salivation and gagging and vomiting. This nucleus is very important in its control of like eating and digestion, and it's also referred to as the gustatory nucleus because of its involvement there. And then again, neurons in the thalamus, they project to many different areas: insular cortex, the frontal cortex, those parts that are involved in taste. There are cells that project from the insular cortex that then carry information to the hypothalamus and to the into the amygdala in particular, because the amygdala, that can be one thing that senses whether a stimulus, such as like a food, is aversive or not. So if you put something in your mouth that's particularly nasty, or if it carries, emotional salience, it's going to be gated through amygdala. And amygdala takes in all kinds of sensory information and processes that information, including taste. So let's walk through the 4 different steps involved in taste, right? So we've got sensory pathways. They all involve these things. With sensory reception, we've got the sensory receptors. Those are the receptors on the taste cells themselves, right? Whether we're talking about the G protein coupled mechanism in umami and sweetness and bitterness, or if we're talking about sodium channels, or if we're talking about proton channels for sour and saltiness, that's sensory reception. Sensory transduction is how that information gets then leads to a change in neuronal membrane potential, which we talked about. Transmission of that information is then sent to the central nervous system via those 3 facial nerves. And then perception is all the other parts, all this stuff happening up within the brain, which allows for us to perceive the mechanism. So the next sense that we're going to go through is smell. Obviously, we don't smell with our noses, right? Instead, we smell with a portion of a part that's inside of the nasal chamber, known as the olfactory epithelium. So when we inhale air, we have a part of our inside of our noses that is sensing the odorants, the chemicals that we can sense, inside there. So let's walk through what the olfactory epithelium is. So the olfactory epithelium is a thin sheet of cells that is on the inner surface of the dorsal surface of the nasal chamber. And it's made up of 3 different types of cells. Now, the main cell is the olfactory receptor cells. These are neurons. They transduce the olfactory signal and send that information to the central nervous system via the olfactory nerve. The cell body of these neurons is located outside of the cribriform plate, which is the bony chamber that separates the brain from our nasal cavity. These olfactory receptor cells, they have these cilia that extend into a mucus layer that is just on the outside of the olfactory epithelium. So all of these cells make up the olfactory epithelium. As I said, it's a thin sheet of cells that's on the surface like this. And then you would see these cilia inside there. There are 2 other types of cells. We have supporting cells. These are necessary for producing mucus. So the mucus here is made by these supporting cells. They do other jobs too. And then there are basal cells. These, similar to the basal cells that are found within the taste buds, these basal cells, they divide and give rise to new olfactory receptor cells. So these olfactory receptor cells do wear out. They do get replaced and changed. And there's changeover. The regeneration cycle in humans is around 4 to 8 weeks. So going over some of the key concepts for smell here, as I've already mentioned this word once, odorants. Odorants are similar to tastants, except that this is in air instead of being in our mouth. Odorants are going to be the chemical stimuli in the air that dissolve in mucus. So they got to be dissolved into the mucus to act on the cilia of the olfactory receptor cells. And the size of the olfactory epithelium actually varies quite a bit across different species. So as I mentioned, in humans, if we're doing a cross section, so we're looking coronally at the nasal cavity in humans, the olfactory epithelium, like I said, it's just going to be on the dorsal portion right here. If we look at it in mice, and we do a cross section, obviously humans, proportionally, this is a bit bigger than it is in mice, but you can see that like more of their chamber has a lot more olfactory epithelium. And then within dogs, they actually have a huge olfactory epithelium. As you can see, it covers just about everything on the inside of the nose, including all the turbinates that, like these bony protuberances that extend out. We have these turbinates as well, as do mice, but we don't really have a very extensive olfactory epithelium around these. But dogs do. They have a huge olfactory epithelium. And what that means is that, humans, and a typical human, despite humans being bigger than most dogs, we only have around 12 million olfactory receptor neurons. A rat, which is a lot smaller than a human, a lot smaller, they actually have more olfactory receptor neurons than a human. And then a typical dog is going to have something like, a billion olfactory receptor neurons. Huge number of olfactory receptor neurons. A bloodhound has even more. And then what we're showing here is the surface area of the olfactory epithelium. So dogs have a much bigger surface area that's inside their nasal cavity that is dedicated to smell. You can see it's huge compared to humans, like 20 times in size, despite the fact that humans are bigger. And even rats have a bigger olfactory epithelium than a human, despite the fact that humans are like 100 times the size of a rat. So humans are not very smell focused. Other animals are. And there's a ton of interesting neuroscience behind this and ethology behind this, like how mice and dogs use their sense of smell, and other animals too. But we're not going to have time to get into it. We're just going to talk about basic underlying neural circuitry that regulates smell. So let's go through this a little bit. The signal transduction that happens within the vertebrate olfactory system, and focusing on these olfactory receptor cells. So olfactory neurons, they have this single thin dendrite that extends to the surface of the epithelium. And within that, this mucus layer, we have the extension of these thin cilia. The odorants become dissolved in the mucus, and then they will bind to the surface of the cilia. This will then activate the signal transduction pathway. On the opposite side of the olfactory receptor cell is a thin, unmyelinated axon, and then this goes through holes within the cribriform plate, going up to the brain, and then this is the olfactory nerve. Now, in humans, there isn't a single distinct olfactory nerve. This is true for most of the mammals. Instead, it's separate. Like there's going to be a bunch of little holes that go through the cribriform plate where some of these axons will bundle and then connect to the olfactory bulb. Now let's talk about the internal mechanism of olfactory receptor cells. So this is what happens when an odorant binds to an odorant receptor. So if we're looking at the cilia, so this would be the cilia within the mucus. Odorant has dissolved in the mucous membrane, and it will bind to an odorant receptor. An odorant receptor protein is a type of receptor that's found within the cell membrane of the cilia. That is then those odorant receptors are G protein coupled mechanisms. So it's going to stimulate the production of G-ULF, right? So this is a G protein that is specifically for olfaction. This is then going to trigger the adenyl cyclase mechanism. Adenyl cyclase is a common downstream mechanism of G proteins. It stimulates the production of cyclic AMP. It converts ATP into cyclic AMP. So therefore, we're going to have an increase in the concentration of cyclic AMP within the cell. This is a second messenger system that then triggers the opening of calcium and sodium channels that are found within these cells. So these channels open. Now we have an influx of sodium and calcium Calcium is also going to bind to chloride channels that allow chloride ions to flow out. So in this case, we have channels that allow chloride to flow out. So this is going to depolarize the cell. So we have massive membrane depolarization because we've got positive ions flowing in and negative ions flowing out, depolarizing the cell, causing action potentials as long as it reaches a membrane threshold. So this is a little bit different than the taste cells in that those have mostly graded release of neurotransmitter. In this case, so they don't have action potentials, these olfactory receptor cells actually act as neurons because they do have action potentials. And in fact, that's shown here. So there's a wide diversity of olfactory receptor proteins, and it can vary an awful lot across different species. So rodents, this is what allows us to discriminate the thousands of different odorants. The number of olfactory receptor proteins, as I said, they vary across species. Rodents can have around 1,000 different receptors within the genome. Humans have only around 350 functional olfactory receptor proteins. There are a bunch of pseudogenes within our genome, clearly like remnants of olfactory receptor proteins that used to work, but now they no longer work. So they just have broken down because we're not particularly olfactory focused as a species go, whereas rodents are. They depend upon their olfaction quite a bit, and so they have a very well developed olfactory system, and that includes the ability to discriminate lots of different odorants that humans cannot. Right? If you have a lot of different receptors, you can discriminate many different kinds of odorants, and humans just can't. So each olfactory receptor cell, this is the other interesting thing about the olfactory system, they express just a single olfactory protein. So there'll be one protein, so one of those G protein mechanisms, G protein receptors in each olfactory receptor cell. And so if you were to record activity when we present different odorants, say like a citrus odorant or a floral odorant or a peppermint odorant or an almond odorant, you might have chemicals that are within each of these that do act on those particular G proteins. And so if we look, for instance, at this green cell, if we're going to look at the activity of this green cell, you can see that this G protein receptor is particularly sensitive to a chemical that's within the citrus odorant that activates this cell quite a bit, and it fires a lot. There's some odorant in here that activates the floral, within floral that activates the receptor as well, but it doesn't fire as much. You can see it fires less. But if we're looking at this blue cell, well, it is also sensitive to citrus, but it doesn't fire as much. And if you look at this red cell, well, it doesn't really fire at all. But this blue cell is particularly sensitive to things in floral, less so. And then this red cell is sensitive to it, but just a little bit. Then if we provide a peppermint odorant, you can see the green cell fires a lot. This fires very little. This fires quite a bit. So this cell is particularly sensitive to peppermint. And then almond has another different combination where this doesn't fire at all, and this fires quite a bit, and this fires just a little bit too. So you can see that like there isn't any one particular type of odorant that's going to be like, oh, this is the peppermint odorant. In fact, we have many different receptors that fire with the same kinds of odorants, but they fire to different degrees and in different combinations. You know, almond, this isn't firing at all, but it's these two that are firing some. And it's going to be the combination of all of this information, not just across 3 receptors, but you know, maybe as many as 350 receptors that tells us I am smelling almond, or some other combination of firing of activity that will tell us I am smelling peppermint. This is known as the population coding scheme. You can see that we get a distinct response pattern depending upon the odorant. And it's this pattern that we see across the different olfactory receptor cells that tells us, our brain, that can then recognize that this is citrus, that this is floral, that this is peppermint, and this is almond. It's the combination of all this information that tells us this. Now, the work on this, a lot of this work was done by Richard Axel and another neuroscientist named Linda Buck when she was a postdoc in his lab. They received the Nobel Prize for this work in 2004. And this is one of their many famous papers on this topic. And one of the figures that I took from this paper showing the olfactory receptor cells. And so, yeah, they identified this mechanism and how it works. Really amazing work. And I've actually had the pleasure of listening to Linda Buck give a presentation. And she's a very dynamic speaker. And it's very cool to hear a Nobel Prize winner give a talk on their work. So let's talk about then how this information gets into the central nervous system. So as we've been talking about, we've got the first order neurons. Those first order neurons are the olfactory receptor cells. These different cells will fire. And as you can see, we've got these axons passing through the cribriform plate. All these little branches. This is the olfactory nerve, okay? So this is the nerve, and this information is carried in to the olfactory bulb. Within the olfactory bulb, we have a second order neuron. That second order neuron is going to be on the other side of these gaps, what are called glomeruli. We're going to talk more about these over the next several slides. These second order neurons then send their axons down the olfactory tract, which goes into the brain. And so this is within the olfactory bulb. And so in humans, this is what it looks like right here. And then this is the olfactory tract. And then all these little branches here, this is called the olfactory nerve. It's a bunch of them, the tiny little ones that stick through. Some species have just a single nerve because the olfactory epithelium doesn't actually have to go through. As you're going to see in tadpoles, they just have a single nerve, not a bunch of little branches like this. But then this is the organization. So the primary nerves, the primary neurons, the first order neurons, they synapse onto the secondary neurons in glomeruli. So let's talk about glomeruli and mapping. So the information across these different types of receptor cells, these receptor cells are going to have a particular odorant G protein odorant receptor that they express. This information will be distributed across the olfactory epithelium, right? So you might have a green cell over here, over here, over here, over here. But this information gets collected in a particular glomerulus. So a single of these little sort of like bubbles, we'll see what they look like in a slide or two. The sensory information for that particular olfactory receptor is collected into a particular place. So we want the information to be collected across the olfactory epithelium, right? Like if an odorant comes over here, we certainly want to be picking it up, or if it's over here, we want to pick it up, or if it's across the whole thing, well, we want to know that all these neurons are firing, but we're going to gather all that information into a glomerulus. So each glomerulus receives input only from receptor cells expressing that particular receptor protein. So this is going to be collecting the green receptor protein. This is going to be collecting the red receptor protein. This is going to be collecting the blue receptor protein. Another important note is that the olfactory bulb is quite a bit different across the different species. So in general, primates, especially the non-human primates, they don't have particularly big olfactory bulbs. You can see it's actually quite small. Whereas animals like dogs and rodents that depend upon olfaction quite a bit, you can see that they have a huge olfactory bulb relative to the rest of their brain, which speaks to just how important olfaction is to some species and how of little importance it is to others, such as humans. So this is an example of a Nissl-stained cross-section of the olfactory bulb in mice. I actually find looking at the olfactory bulb in animals to be quite beautiful. It's really quite a striking organization that we see. And yeah, so this is looking at the different layers using Nissl staining. And here are the glomeruli. So this is the glomerular layer. This is the outer portion of the olfactory bulb. If we do a cross-section through here, there's another layer known as the outer plexiform layer. These are the mitral cells. These are going to be the primary second order neurons. We'll get into how they're connected in just a moment. Then there's another separate layer called the inner plexiform layer. And then we have the granule cell layer. This is just loaded with neurons. Okay, so there's tons of neurons in here. So you can see that there's a ton of neurons within here. We've got the mitral cells that are just on the outside like this. And then we've got the glomeruli out here in the glomerular layer. And you can see these empty spaces. So these are largely free of cells, right? You can't see too many cells in there. There are cells in between the glomeruli, which is why it stands out like this. But each glomerulus is going to be collecting the information from olfactory receptor cells that carry the same receptor protein. And then they're going to be connecting to, I think one or a couple of different mitral cells that'll be sending their dendrites here. So in fact, let's take a look to see what that looks like. So as you can see, you got these olfactory receptor cells. They are then synapsing onto mitral cells that send their dendrites into the glomerulus. So these empty spaces, this is like white matter, basically. Like if you don't see in a Nissl stain, if you don't see a lot of blue dots, that means that there aren't cells in there, like the cell bodies are not in there, but it is filled. These aren't empty. They're filled with dendrites and axons. So that's why that these are empty. If we could label the dendrites and axons, we would just see it would be just loaded with dendrites and axons. But since we're labeling, this label is to label the cell bodies, that's why it looks empty. So the information would be coming in this way and connecting there. So these are the important parts to know when we're talking about this. Okay, so there's a lot of different cells in here. I don't want you to have them all memorized, but what I do want you to know are the olfactory receptor cells and how they are organized, how they synapse into particular glomeruli, that they connect to mitral cells, which is going to be the second order cells. And then we're going to be talking about the granule cells as well, okay? So the granule cells are also an important regulator. The other thing is that, you need to get flexible with how you think about the brain, right? We're looking at the dorsal side here, right? So this would be the dorsal side of the olfactory bulb. So this is in reverse order to this. So this is not matching this. I have it side by side here, but you have to think of it as being flipped around, right? So this would be the glomerular layer here. This would be the outer plexiform layer here. This would be the mitral cell layer here. There's the inner plexiform layer, and then the granule cell layer would be right here. Okay, so you got to be flexible with your brains and thinking about this. Okay, so mitral cells, as I said, they are the second order projection, and they actually project to tons of different cortical areas. So those mitral cells, they have axons that spread all over the place. So this is a cross section of a brain. We're looking at the olfactory tubercle, piriform cortex, entorhinal cortex, and amygdala, all in one section. And what I want you to, and then what we did is we filled mitral cells. So we filled the mitral cells to see where they project. So this is just showing the different brain areas. Then this is showing the same thing, but now we're illuminating those mitral cells and where they project. And as you can see, they project to a lot of different places. The olfactory tubercle, the entorhinal cortex over here, the piriform cortex, you can see it gets tons of information, and as well as the amygdala. The amygdala is getting hit with all kinds of information. This portion of the amygdala right here is getting lots of olfactory information. So there's lots of places that receive inputs from the olfactory bulb. So let's look at this a little bit more and a little bit more of a schematic pathway. So olfactory receptor cells, they synapse onto mitral cells, right? That is the first order to second order neuron. Olfactory receptor cells synapsing onto mitral cells. Those mitral cells send projections to all kinds of different areas, as I just said. Olfactory tubercle, entorhinal cortex, amygdala, and piriform cortex. Now, the interesting thing is that we have a projection from a sensory modality directly to cortex. The entorhinal cortex, olfactory tubercle, piriform cortex. This makes smell unique in that it does not go through the thalamus. So there is connection to thalamus. So eventually the olfactory tubercle sends its information to the thalamus. But we do get direct information from the secondary order neurons directly to the cortex without having to be gated through the thalamus. And the thalamus kind of serves as a gating pathway of taking sensory information and then relaying it up to the cortex. And it does do that to some degree. That is part of what is happening here. But But there's a ton of olfactory information that does not go through the thalamus, and that makes smell sort of unique. It makes it. It's thought that sometimes smell can trigger memories that like maybe we are sort of elusive, that we don't understand why like a memory is being triggered. And it might draw up some sort of memory, and you might have like feelings of deja vu around smell. And you might have a difficult time placing it, but you're like, oh, I know I had smelled this at some point. It's thought that like because it's not gated through the thalamus, that it makes those memories sort of strange in some ways, and that's why smell sort of is put into a special category than the other senses. So again, this is an illustration of the connections that happen. So we've got the olfactory receptors, synapse onto the mitral cells in the olfactory bulb. This is the primary olfactory nerve. This is the olfactory tract that connects then up to the major targets, piriform cortex, olfactory tubercle, amygdala, and entorhinal cortex. And then information from there can connect to a bunch of different places. As I said, there's thalamus, there's orbital frontal cortex, hippocampal formation that receives input from the entorhinal cortex, as we're going to get into. So we can have, entrainment of memory to the hippocampus. So there's a bunch of different, smell is an important thing, especially for animals that really depend upon it. And it's used to influence motor control, our emotional reactions to odor stimuli, reproductive behavior that depend upon, the olfaction of pheromones, for instance. Things related to feeding, things smell good because they taste good and you need to eat it, or things smell bad, you want to avoid it. So, even humans depend upon this a lot. A lot of this gets gated through the amygdala, and we're going to talk about that later. So we'll get to it. So for now, we don't have to worry about that so much, but we will get into it. So there are many different ways to measure neural activity in live animals. We've talked about various ways. We've talked about doing electrophysiological recordings of individual neurons or groups of neurons using either intracellular or extracellular electrodes. This is, the famous experiments with cats to determine bar orientation and the visual cortex. Within the visual cortex, we also talked about like movement artifacts within this part of the brain called MT. And some of this work has been done in humans, where the MT neurons will fire when humans are having to look at, say, fields of activity. They're in an MRI machine like this. They're looking at a screen that's showing dots either moving or they're stationary. And if they are moving, MT tends to fire. You can use functional magnetic resonance imaging to look at groups of neurons that are firing. So these are two main ways that you can look at neural activity in animals. Arguably, these are probably the two most common ways, but there are other ways, and we're learning more and more about how this works, and it's being used in more and more types of animal models. So I want to spend a little time talking about that in the next slide. So we can measure neuroactivity in live animals using what's called calcium imaging. So with calcium imaging, what we're doing is we're monitoring intracellular calcium. When neurons fire, there's a brief increase in intracellular calcium concentration, as we've been learning about, right? So if there's an action potential that occurs, so if we're just monitoring membrane potential, during that action potential, what we see, if we could monitor calcium concentration, we're also going to see a sharp increase in calcium concentration that lasts for a good long while, several milliseconds, tens of milliseconds. There'll be a brief elevation of calcium within that cell if there's been an action potential. That's because we have a lot of calcium on the outside of the cell, and we have a lot of calcium on the inside of the cell, but specifically within the endoplasmic reticulum. If we have an action potential that's occurred, well, those calcium channels open, and then we have an increase in the concentration of calcium. You can see a lot more of these ions are located in the cytosol of this cell, but all that excess calcium starts to get pumped out as soon as it increases, right? So we have the sudden influx of calcium, and then it starts to get pumped out of the cell, and then also taken up into intracellular calcium stores into endoplasmic reticulum, as well as the mitochondria take up excess calcium. And then eventually the concentration gets back to baseline, and you can monitor this, which is interesting. we can use genetic tools such as GCamp. This is a genetic tool that has been developed that has a fluorescent molecule called enhanced green fluorescent protein that is calcium sensitive. It has a calcium calmodulin sensor attached to it, and when calcium binds to the calmodulin, this molecule then binds to this, which then changes the structure of the green fluorescent protein, making this fluorescent. And so when you shine a light on it, it will then emit a light in the presence of calcium. So if calcium is here, this can glow. If there is no calcium, it won't glow. So it's present in all circumstances, and you can make cells express this. This is just a protein. This is a protein attached to this protein, which is attached to this functional unit here. And when calcium binds, there's a conformational change that allows this then to fluoresce. But in the absence of calcium, these two things are disassociated, changes the structure of this, and that makes it inactive. Super cool technique. And let's look at an example of what this looks like. So essentially, we can make cells within cortex, olfactory cortex, express GCamp. And so these cells have GCamp in them. And if they start to fire, they will start to glow. And what we see when we do these kinds of experiments, so if we put in GCamp into a portion of the brain, where, like let's say we're going to look in the piriform cortex or the entorhinal cortex, or the olfactory tubercle. So you can inject a virus that allows for the overexpression of a GCamp, and then we look at all the different cells that are there. If we provide some kind of odorant, like say this is alpha pinene, this odorant, this is the chemical structure here. It is, when we smell it, we can kind of refer to it as being piney or resinous. It's basically what gives pine its pine smell, alpha pinene. Or a different odorant, such as octanol. This is an odorant that we typically find in fruits, and it's associated with being fruity or citrus. Or hexanol, cut grass, versus octanol. Okay, so those are the two different combinations of odorants. If we provide pinene, we can see within portions of the brain that are odor sensitive, such as like parts of piriform cortex. If we do pinene, some cells will fire. If we do octanol, other cells will fire. But then if we do pinene or octanol, sometimes some of those cells also fire to both of them. So it's going to be it's population coding here. So if we provide pinene, it's going to be the green cells and the yellow cells that are firing. If you do octanol, it is going to be the red cells and the yellow cells that are firing. Yellow is an indication that these cells are sensitive to both pinene and octanol. And we can tell this in a whole population of cells by using cyclic JMP. So this is what this looks like. This is actually a live image where an odorant is presented, and then there's been firing that's occurring, and we can monitor, well, these cells have fired in response to the odorant being presented. And then if you provide both of these odorants at the same time, then you're going to see the red, green, and yellow cells fire. And they will definitely fire. And in fact, it might be the case that the yellow ones have an even stronger response because these cells are going to be sensitive to both pinene and octanol. Now, it's likely that these are acting on different receptor proteins. So therefore, it's going to be different glomeruli that take in this information. And then this information is then recombined in cortical areas to give us a sense of, oh, this is a piney resinous smell, or this is a fruity citrus smell, or you're smelling both of these at the same time, right? You can have both of these in odorants being presented at exactly the same time, and we may be able to distinguish those two because of the population that gets presented. And then if we present octanol again, but with hexanol, well, you can see that the neurons here, and again, the gray is indicating cells that are not firing at all, but here we have different cells that are firing, right? Hexanol is triggering these two cells to fire, which are not firing to any of these. We have these cells that are firing, but not here. And then this yellow cell is sensitive to hexanol and octanol. as well as alpha pinene. Right. So it's going to be this combination of information that tells us what kind of odorant that we're picking up. And it's a population coding type mechanism that allows the brain to do this, to tell us that this is what the smell of cut grass is. The red cells and the yellow cells are going to tell us that this is a fruity citrus smell. And then we can even tell the difference if we smell both of these things at the same time. It's going to be both populations of cells that are firing at the same time. So as far as the four populations, the four steps of olfaction, we've got sensory reception that occurs within the olfactory receptor cells, acting on those G protein odorant receptor proteins that are in the cell membrane of the olfactory receptor cells. That triggers action potentials within the olfactory receptor neurons. That information is carried to the olfactory bulb. That gets transmitted to the brain via the olfactory tract into the cortical areas that allows for perception. And then there's further information that can be gleaned from olfaction information in the higher order portions of the brain. So that's taste and smell. If you like this kind of content, please like and subscribe, and I will talk to you later. Bye-bye.